Hi, let's talk about neurons and how they work and how signaling works and action potentials. So neurons have two ways to communicate. One is electrical, which is action potentials, and the other is chemical, which is specifically synaptic communication, which we already talked a little bit about that in a previous chapter. Signals from the sensory inputs have to be interpreted, and then motor responses come out of the central nervous system. And there are two places where sensory signals and motor signals can be integrated. One is, of course, the central nervous system, but the other is also ganglia, or a group of neurons. Most of the neurons' cell organelles, like the nucleus and most of the mitochondria and the endoplasmic reticulum, are in the cell body. Neurons also have dendrites. Dendrites are where they receive signals from other neurons. And axon, or usually one axon, is where they transmit signals to other cells, which could be other neurons or muscle cells. And where the action potential is initiated, the beginning of the axon on a neuron is called the axon hillock, and that's where we see sometimes chemical signals converted into an action potential. So here is a neuron here, and neurons are quite large, as we saw in lab when you were looking at those under the microscope. The cell body is quite large, and it's manufacturing a lot of molecules that are used by the rest of the cell. There's a lot of membrane in neurons. So dendrites are where they're receiving signals, and one neuron could be connected to 100 other neurons via dendrites. And then they typically have a single axon, and that's how they transmit signals to other cells. These connections can be synaptic terminals that are chemical in nature, but they can also have electrical connections between two neurons. And we talked a little bit about synapse. We're going to talk a little bit more in detail about how neurotransmitters are released in the synapse. So the chemicals that are released, which we talked about one already, acetylcholine, are neurotransmitters, which allow an action potential to be turned into a chemical signal and then back into an action potential in the next neuron. There are a lot of other cells in the nervous system besides neurons. There are support cells. And these support cells generally are called glia, or glial cells. And there are quite a few different types. And they are much smaller than the neurons. So in this particular a colorized microscope image here, you can see large neuronal cell bodies, and then much smaller cells. Those are the glial cells. And there are different types, and they have different functions. Astrocytes, oligodendrocytes, and microglia. And microglia, you might imagine, are small. These are found in the brain. Ependymal cells, radial glia, Schwann cells, satellite cells, and enteric glia are in the peripheral nervous system, and we'll talk about the functions of some of these in this chapter and some in the next two chapters. And these cells really allow the neurons to do what they do. Neurons have very high metabolic activity. They throw off a lot of waste products that are not removed rapidly enough, and that's one of the functions of the glial cells, especially in the brain, is the removal of waste products of the neurons. Some of them, like Schwann cells, also provide insulation and allow action potentials to be transmitted more rapidly. Some of them are part of the blood-brain barrier, which we'll talk a little bit more about. Sensory neurons, which we'll talk more about in, I think it's chapter 50, receive our input from the outside world. We have sensory neurons that can detect cold and heat and light pressure and deep pressure and light, as in our eyes, and smell and taste. We have interneurons. These are in the central nervous system. These integrate and generate a response to stimuli coming in from the sensory neurons. And then motor neurons attach to muscles. Motor neurons go from the central nervous system to muscle cells. And they have different shapes, and you can see these are very, generalized shapes. So sensory neuron, the cell body is outside the central nervous system. Dendrites, again, are where they are receiving their signals. So these dendrites could be in the tips of the fingers. Some sensory organs are just naked dendrites of a particular neuron. And then the signal is transmitted away from the cell body along the axon. Interneurons receive information from other cells, send out information to other cells. They are in the central nervous system. And motor neurons, again, they're receiving information and transmitting it to muscles. Motor neurons, the axon of a motor neuron ends at a muscle cell. And it could be smooth muscle or striated muscle. So, and as an example of a response that requires integration in the central nervous system, we have a little carnivorous snail here, a cone snail. Cone snails paralyze their prey and then eat them, and they detect their prey by constantly sampling the seawater. Cone snails are marine. They sample the seawater, detecting chemicals that are released by fish. When they receive the correct input that a fish is near, they respond with this little barb that they send out that will stab the fish, inject it with venom, and paralyze it. There's a nice little video here that I've linked to of a cone snail, but I can't embed it in here because it will be a copyright violation, because I think this one is a National Geographic video, but I'll put a separate link to it in our playlist. So the central nervous system is the brain and the spinal cord, and the peripheral nervous system is everything else. So all the nerves in the whole rest of your body is what we call the peripheral nervous system. And nerves are bundles of neurons in the peripheral nervous system. So when you get to anatomy and you learn where various nerves go, like down here around your elbow, a nerve is just a bundle of axons from particular neurons. This video has no sound. It's just a little three-dimensional video of an injected neuron so that you can see the three-dimensional structure of it. It's been injected with a fluorescent tag, probably another microtubule fluorescent tag, since the dendrites and the axon are full of microtubules. That's where a lot of their structure is coming from. Okay, so action potentials. This is an important concept that you're going to use in future classes, so make sure that you ask if how an action potential works isn't clear. This is one of those concepts that the first time you hear it, it sometimes seems very confusing, but once you get it, it just, it just makes sense after that. Okay, so we've talked before about the sodium-potassium pump. This is, it uses ATP to pump sodium out and potassium into the cell. So we also get a charge imbalance because it pumps out 3 sodiums for every 2 potassiums, and each has a charge of +1. So we end up with a positive charge outside the cell and a net negative charge inside the cell. That's called the membrane potential or the resting potential. And this is essential, especially for neurons, because without this resting potential, no action potentials are going to be possible, which means that the neuron cannot communicate with any other cells. So this a wave of a flipping of this membrane potential, so that instead of the positive charge being outside the cell, for a millisecond, the positive charge is going to flip to inside at a particular spot on the membrane, and then it's going to flip back. That's the electrical signal that neurons are using to communicate. That's what we call an action potential. And so the sodium-potassium pump is running continuously. It doesn't stop. It uses up 55% of the brain's total consumed energy. And the brain is a very expensive organ that uses a lot of your total calories for your body. So, and other cells in your body, not just neurons, are using the sodium-potassium pump. So this is using a huge amount of energy, and it's just constantly trying to restore that resting membrane potential. And you can also see why having the correct amount of sodium and potassium in your body is also really important. Without that, the membrane potential is going to fail if you don't have the correct amount of sodium and potassium. So if you remember back from the membrane stuff, besides facilitated diffusion, we also have channels that are not always open, gated channels. So a gated channel in the membrane opens in response to some kind of a signal. And when it opens, ions can just flow down their concentration gradient. So things are going to go from a high to a low concentration, and then the gate's going to close again. So we have voltage-gated potassium channels and voltage-gated sodium channels in the membrane. And that is what is going to generate the action potential. First, the sodium-gated channels are going to open, or the voltage-gated sodium channels are going to open, and sodium is going to flow into the cell, and then that's going to reverse the membrane potential, and then the sodium channels are going to close, and the potassium channels are going to open, and potassium is going to flow out, and that's going to restore the membrane potential. So that's an action potential, thanks to those two types of voltage-gated channels that are going to open in response to changes in the membrane potential. And not in the whole cell, just in a particular spot on the membrane. So in gated channels, they could allow ions to flow down their concentration gradient. Gated channels don't use ATP. Things can only flow from high to low concentration, but only when the gate is open. And they can open when they can be a receptor, and they can open when a particular molecule binds to the gate. That can open it, or they can open in response to a change in the membrane potential, and that would be a voltage-gated channel. The membrane is normally polarized, so it normally has an excess of positive charge on the outside and negative on the inside. And for neurons, the normal voltage, the normal resting potential, is about minus 70 millivolts. That's the resting potential that is generated by the sodium-potassium pump, and none of the voltage-gated channels are open. We're going to talk about hyperpolarization and depolarization. So a depolarization means that it's going towards zero. So zero here would mean that there's the same amount of positive and negative charge on both sides of the membrane. There would be no membrane potential there. That would be zero. So depolarization means we're heading towards that. Hyperpolarization means we're going the other way. We're going more negative here, more away from the threshold potential that's going to trigger an action potential. So we have our resting potential here. And if depolarization, or if hyperpolarization is occurring, so hyper means it's going to be going away from triggering an action potential. So when a neuron is receiving input, that input can have one of two effects. It can cause depolarization or it can cause hyperpolarizations. So if it's causing hyperpolarizations, that means it's causing the cell, or that little part of the cell, to be more polarized. It's going away from the threshold potential where an action potential is going to be triggered. Signals that are causing hyperpolarization are triggering more potassium to diffuse out, making the inside of the cell even more negative. That's a hyperpolarizing signal. A depolarizing signal is making this is taking the cell closer to an action potential. A depolarization means that sodium channels are opening and it's taking the cell closer towards the threshold potential. Lacey, stop. That's going to make it more likely that an action potential is going to be triggered. So when this, let me go back to my pointer here. So when we get to the threshold potential, that's the potential at which the voltage-gated sodium channels are going to open, and that's going to trigger an action potential. So a depolarization means we're getting closer to triggering an action potential. So these are graded potentials, meaning they're different magnitudes. So one neuron can be receiving inputs at different points that are either taking the cell closer towards firing an action potential or depolarizing or hyperpolarizing and taking the cell further away from generating an action potential. And they can be additive to one another. Hyperpolarizing signals and depolarizing signals can cancel each other out and have no action potential result. So an action potential down the axon is going to be triggered when the membrane potential depolarizes to the point where it reaches that threshold potential. Then all the sodium gates are going to open and it's going to trigger an action potential. Before that, we could have small changes, you know, some hyperpolarizing signals, some depolarizing signals, but there has to be enough of a depolarizing signal. When we get to that threshold, all the sodium gates open, and then that triggers an action potential. So an action potential is, we could have graded signals. Finally, the threshold potential is reached. The voltage-gated sodium channels open. Sodium rushes into the cell down its concentration gradient. Remember, these gated channels aren't pumping things across the membrane against their concentration gradient. They're just allowing things to flow downhill. They're just opening the dam and letting things flow downhill. That's an action potential. And this depolarization, that's going to trigger the potassium gates to open. So sodium gates open, the charge is reversed. See, now we're in the positive here. That's going to trigger, the sodium gates are going to close and the potassium gates are going to open. And that's going to reverse the potential. And then there's a period here where another action potential cannot occur while the sodium-potassium pump restores the membrane potential. So that's one of the reasons that the action potential only goes in one direction, because there is a refractory period for any particular part of the neuron after an action potential goes by while the sodium-potassium pump resets. The sodium channels will not react immediately after an action potential has gone by. There is a refractory period where there cannot be another action potential.
This baseball player springs into action, his nervous system rapidly processing information in a network of nerve cells, or neurons. Each neuron receives input from one or more cells. In response, the neuron may generate an electrical signal known as an action potential that travels down the length of the axon. Let's take a closer look at the plasma membrane of an axon. Even without an action potential, the axon is a busy place with many ions moving across its membrane. Much of this ion movement is driven by the sodium-potassium pump. Using energy from ATP, sodium-potassium pumps actively transport sodium ions out of the cell and potassium ions in, creating an uneven distribution of charge across the membrane. Some potassium channels are open all the time, allowing potassium ions to leave the cell. As a result of these ion movements, the inside of the cell is negative relative to the outside. This condition is called the resting potential. The membrane of an axon is also packed with gated ion channels that open and close during an action potential. At resting potential, the gated channels are closed. If a stimulus changes the distribution of charge across the membrane sufficiently, the gated sodium channels open. Movement of sodium ions across the membrane makes the inside of the cell more positive. This reversal of the charge distribution causes the gated sodium channels to close and the gated potassium channels to open. As potassium ions move out of the cell, the original charge difference is re-established across the membrane, closing the gated potassium channels. This sequence of events is called an action potential. The sodium-potassium pump restores the distribution of ions back to their previous levels at resting potential. Let's look at how the action potential is conducted along the axon. As the change in charge difference across the membrane spreads from open sodium channels, other sodium channels farther along the axon begin to open. The original sodium channels close, and adjacent potassium channels open. As potassium ions move out of the cell, the original charge difference across the membrane is restored, and then the potassium channels close. Meanwhile, new sodium channels open, followed by the opening of new potassium channels and the closing of sodium channels. In this manner, the action potential is propagated along the axon of the neuron, eventually reaching another cell. The information carried by this action potential will be processed with other information, permitting this baseball player to make a spectacular catch.
So, where the action potential begins is not on the cell body of the neuron, but it's usually let me go back to my pointer here. It's usually on the axon hillock where the depolarization begins. And it's from all the different signals that are being received on the dendrites that kind of add up spatially, and they add up over time to a depolarization event that reaches the threshold potential and generates an action potential. So let's look here. So here's an axon. So the whole thing isn't depolarizing. It's just one little portion. So an action potential hits one part of the axon, comes down from the cell body. So the dendrites on this axon are receiving signals from other neurons. Some are depolarizing signals, some are hyperpolarizing signals. Eventually, they will add up to have this region of the cell reach the threshold potential, and that's going to trigger an action potential down the axon. So once that threshold potential is reached, the voltage-gated sodium channels open, sodium rushes into the cell. That is going to trigger the same reaction in the neighboring plasma membrane. And that action potential will then move down the axon. That depolarization event, changing the positive to being inside the cell and negative to being outside the cell, that's what's going to open the voltage-gated potassium channel. So now the voltage-gated potassium channels open, potassium rushes out of the cell, again, just going down its concentration gradient. It has nothing to do with the charge. It has to do with the concentration of potassium. That, however, now that we have sodium inside and potassium outside, that's going to restore the negative membrane potential. And that's going to also be the cause of the refractory period. The sodium-potassium pump is going to now restore sodium on the outside, potassium on the inside, so that another action potential could move down that same axon. And it's this refractory period that is the reason that action potentials don't move in both directions. Why don't they move in both directions? They don't move backwards because of the refractory period. By the time the refractory period's over, the neighboring portion of the axon has already restored its membrane potential, so there's nothing to trigger another action potential unless it's coming from the cell body again. So that's how action potentials move down the axon. Okay, in the next section we'll talk more about synapses and the additive effects of different signals being received by a particular neuron.